All right, so I'm going to go through the high throughput, focus on it a little bit, but so again, for those of you who joined later, we had a couple stragglers who are experts and didn't need the intro section. So welcome to you, the experts there. Again, I just want to emphasize we're not interested in a performative workshop. We want to make sure it's useful to everyone. Please come and go. I'm going to go over the HT Sacks for, hour-ish. Maybe I'll cut it a little short just because I ran too long with the other one. Um, Mikkel will go into more detail with the SEC Sacks and tools after me.
I
Uh,
would just mention, Greg, if I may say something, what the schedule is, I will again focusing on how to analyze the SEC SAC rather than going at cases.
okay. Then we'll have a little segment for you to hear from your fellow Sybil's users and sufferers of our Beamline. See what they're trying to do, and maybe there's some good connections there. The online doc we've mentioned. I was going to throw that link into this spreadsheet, but you've got it in the chat. Maybe Amici, if you're still there. Oh, here, I'll just do it. Let's see. Anyway, Catherine or Amici, could you throw it in the chat again? I think the
Sure.
challenge is that the people who came later don't have the chat record. So if you throw it in again, everybody who's here will get it again. And then we're going to respond to that document later in the afternoon. We gathered information from the survey. Thanks all of you guys for filling that out, those of you who did. This is the breakdown. Again, thanks also, Catherine, for organizing this, of who you are. Not many of you do only HT Sachs. There's a small slice, and that's how I've kind of changed it. Many of you do both, and a lot of you have never collected Sachs data before. So just so you know who's in the Zoom room. Um, your success is very important to us. We spend a lot of our effort making the facility useful. All the tools that you see, we want them to be useful. We're not just trying to do cool stuff, right? So we have a stake in your success. And so that's why we're holding this workshop, right? We get hundreds of projects every year, but around 30 papers per year. So we'd like to increase that success rate. Um, we want to know what you do with the data you collect, and we want to make sure that you use the data to its maximum advantage, not just plotting, RG as a function of salt. Um, the workshop is motivated by the ALS schedule, and for those of you who um, have not uh, gone through the process of registering to apply for time, this is what you'll encounter. And we've just released dates. So these are all, last data collection was June 24th. Uh, the next official collection is October 14th. And yellow is SEC SACS and green is HT SACS. And between October 14th and Christmas time, there are not many shifts, right? And so we have, it's September 12th now. It's going to take you guys some time to prep samples. We've given you basically a month of time. So we're hoping that this workshop triggers for you some action to get you starting the planning. We don't want you coming in here on October 14th and then starting to plan and then samples come, around Thanksgiving time. We want you guys thinking about it now. Generally, publishable data, if you're brand new to SAXS, it's going to take you 3 tries. You're going to send us some samples, and they're not going to work out. There's like buffer subtraction issues, or the column was, the concentration wasn't good, or whatever. Something generally goes not perfectly great. You get some good data, but not everything you wanted. The second time you're prepared, you get awesome data, and this is where most of your data comes from. But then, dang it, I missed some controls. And so that's your third time. You're like, oh, the data told me this, but in order to prove that, I need these controls. So that's where the third time is. So mapping that out through this limited schedule is a challenge. We want you guys starting to think about it now. What was just announced, fortunately, is that after Christmas, there's going to be a 6 months of light, maybe January through July, where again we can crank, and we hope that you guys help us crank. There is an ALS upgrade looming, and I'll show you what that looks like. So the entire ALS is getting upgraded, and they've given us these are the dates: 2025, 2026, etc. And they're changing exactly how this is going to be mapped out. But there is going to be a 1 year dark time sometime, and it could be in 2028, or it could be as soon as July 2026. That's kind of the situation we're in. So what we're trying to do is make the most of Epoch 1, which is the section before dark time. We're trying to plot this out as best as we can so that we're very active. During Epoch 2, which is what we're calling the dark time, we're planning to obviously not just go on vacation and stuff. We're hoping that we're going to upgrade hardware at our beamlines. We're going to help collect at other synchrotrons, and we're negotiating some time with other synchrotrons, and we're going to continue developing our software and beamline controls. So you might ask, what's the ALSU? These are the before and after of what's going on. It's very subtle, but there's a second ring. So there's two rings that are inside the synchrotron here, and that is to hope to convert the current beam, which has a horizontal width to it, to something that's much smaller. And here's the inside of the ring. There's, this is as it currently stands, and then there's these two rings that are coming in. And that will increase what they call coherent brightness for ALS to be the most coherent in the sort of, this range. APS just opened up. They have completed their upgrade. And so that will be also a fantastic resource for you all. For Sybils, I showed you a picture of how it is now. And what we're doing is we're decommissioning the crystallography end station, and we're going to put in this gigantic flight tube. We're going to be able to move our detector. Right now, it's a static 2 meters. We're going to be able to move our detector around inside the SAXS beamline. And we're also going to improve the optics. So right now, our beam profile looks like this at the sample and at the detector if it were pushed 4 meters back. This is 2 millimeters. And none of your samples come in a 2 millimeter wide format. I think this is actually 4 millimeters, sorry, 2 millimeters each side. What we're going to move to is these tiny, tiny beams that have all the photons in it. So basically for us, we have to clip the beam down to, a fraction of its size. We're not using all the light that's coming in to be able to hit your sample and not hit the sample container, etc. So we're going to be putting it all in one spot, and that allows us to do a lot of like time resolved and other things. Another thing that we're exploring is a partnership with another SAXS beamline. So right now, if you go to most SAXS beamlines, you'll see that they do everything. They'll have microfluidics, they'll have some material science in there, they'll have grazing incidents, they'll do time resolve, they just do everything. But we like to do high throughput experiments because we know that biology is mutatable and sensitive to conditions and truncatable and all that stuff. So it does not allow us to do these higher end experiments that we would like to do sometimes. You might have some special case. And so we're looking at a partnership with another beamline that's getting built to be able to do biological experiments on these like crazy experiment, one-off, like let's say high pressure, a sample under high pressure. And that's more interesting than you might think. And then keep Sybils in high throughput. We're also looking at, again, there's so many techniques around the ring. This is cells. There's a symbiote in here, and it's watching the cell split with the symbiote. So this is X-ray tomography, and this is a big, thick cell that you couldn't do with EM. So kind of bridging between these kinds of events and the things we're looking at with SAXS. The Sybil staff, this is the sum total of the staff. This is John Tanner, who started the beamline. Got to give credit to John. He's now at MD Anderson studying cancer. but he started it and give him credit. This is the crew that you've seen. You've seen Amici, you've heard from Scott, Mikhail, and myself, but we have fantastic postdocs, and Josh, you probably interact with a bit. Nazar is mostly doing a lot of biochemical work, but he does have an SPR instrument that he's looking at transient interactions there and combining that with SAXS. So if you have that interest. And then Susan, Catherine, you've heard from. James is at the crystallography. Susan can give you a lot of good advice on all kinds of analysis and sample prep. Brandon helps with our engineering. Antonio is brand new, but he is our lipid nanoparticle guy. And if you have questions for lipid nanoparticles, he's the guy. And then we have a lot of alumni for those of you who are early career and looking at National Lab. Maybe it's a different feel for sure, but we've got a lot of great successful alumni that went to companies and other synchrotrons around the world. So if you're considering a career, it's not a bad thing. Okay, so you'll notice that your data collection is free to you, and that's because funding's been requested for you by these big user facilities. That ALSU project is a $1 billion project, $1 billion. So it's a huge, huge project and a huge investment. If you do come and use the ALS after ALSU is built, they would like to know about it, and especially in their acknowledgments. So they ask you to put an acknowledgement in there for the synchrotron. Same with the ALS now. Somebody built it 20 years ago. They want to get credit, so they want you to put things in acknowledgments. Those people that you saw on the previous slide, are funded by these 4 grants. One is to help general users from NIH. One is to help general users from DOE. And then these 2 are sort of smaller projects, one on viruses and one on DNA repair. So this is just that. So we would like you guys to cite these 2 grants when you use our data. This is what our funding agencies look to. And there's we have it on our website where this is. When we try to know when you publish and you haven't told us, and we'd love for you guys to email us when you've published something, but we do a search for this number to try to pull up everything that's used our beamline. When our funding agencies ask us for that information, they ask it for us all the time, and they threaten our funding if we don't have enough. So, and if you're doing anything with viruses, there's another grant that and we will help you very much if you're doing viruses. We'll help you analyze your data, or if you're doing a cancer project, we will help you analyze your data. And then if we do, we would ask you to cite these 2 additional grants. But these got a bunch of people and investigators in there. This is our main web page. I guess the civils.als.lbl.gov. Right there, sorry, there it is. And we've got a bunch of tabs there, and hopefully we've made it clear to reserve a mail-in time slot. Now, we understand that the application for time is super confusing. There's so many accounts, and there's a ALS account, there's a Sybil's account, there's a simple scattering account. It's a complete nightmare. We understand it's a nightmare, but there's a reason for it. Like, the ALS is a separate entity. They want credit for what you do. We need your login for the ins and outs of data. And then simple scattering is a web depository that is hosted offsite, like at Amazon and stuff. And so they don't have our LDAP information. So anyway, we apologize for that. We understand it's annoying. And we're doing everything we can to resolve it. All right. So this is the high throughput SAXS. I'll go through the movie first. This is the needle picking up data sample and delivering it to the sample environment. And data is collecting. There's a laser beam there instead, so we could do light activation and X-rays at the same time. And then it's being deposited down. And then it's gonna wash. So you see liquid dumping out. And then it's rinsing, it's taking up ethanol, dropping it in and out. And then after this, it will go to a dryer. I guess it's spurting whatever's left out. Oh yeah, there's a couple steps in there for processing. And that's actually a UV measure. If we still had that going, we don't right now. And so this is the hair dryer, and you can see like air being pulled in and out. So that's the high throughput robotics, and I'll go through a little bit more detail there. This is a layout of the beamline, so you can see all of the equipment that Mikkel was just talking about, the HPC, SEC material and the malls. Then we have this robotic system. We've got our 2 meters of sample to detector distance, and then our detector. And this is in real life. SEC, malls, liquid handling robot, path, and then the detector. All right. So we have a kind of a description of how you would do HT and SEC. They're pretty much the same. There's a couple slight differences. We've done a lot better with the SEC SAXS in terms of fair data. So the moment that you start entering stuff into, I want time, this is the column, this is how much molecular weight it is, that data starts getting passed along through the whole process until it's deposited into simple scattering. So you don't have to fill out that information again, and you can access the data in simple scattering. SEC SAXS, we haven't quite done that. This is still, you can access your data by file transfer to our systems. But the idea here is you become a Sybils user, you register with the ALS with a separate account, and this is basically a vestigial process. I mean, they handle this, the safety profile of the system, but this is, you know, this is kind of a broken thing for us. But we really want you to do it because the ALS needs it, again, because it has to justify its existence. We have our own. You book a slot, you prepare your 96 well plate, you ship it or hand it to us, and we collect your data. We process the data to some extent, and then you file transfer your data, and then we hope you cite it. The slight difference here is that you start entering information that becomes metadata that gets passed along until it goes into simple scattering, and then there's simple scattering where you can pull your data from, and that's always accessible to you. Let's see. So setting up your experiment is not trivial. It takes a lot of material. It is time consuming. So we appreciate that. So this is me setting up a plate of GFP and various salts. You can see the GFP and different concentrations. High throughput automation can be a high throughput waste to sample if it's not done carefully. We've been trying to make sure nobody wastes their sample. Here's just a little more detail on the collection. Let's see here, I'll start all of these things. So you can see a timer up here. This is going through a plate. Robots coming through, picking up samples from the plate. You'll see the needles. These are the landing in the sample cell. We're shooting through the needle through this window. This is GFP, and sometimes you can see a gray material here. That's radiation damage that after exposure that's happening. And then, you know, you get your SAXS profiles. This particular is GFP is highly charged, so it's repulsing itself from each other, so you get this downturn in the small cube. Yeah, and that's how we get quick analysis. Let's see if I can step through. Okay. For those of you collecting high throughput for the first time, there's things to be aware of, right? That you want to know. There's this washing step. When was it washed? Is there cross-contamination possibly? If you've got issues, we want to know about them. We use partially the liquid, the system liquid, which is water, to wash, but there's also an ethanol step in there, and there's a drying step in there. And again, we're shooting through the edge of the needle. how you set up your sample, and you got different things you want to do. You might want to do some construct screening. And so you might have one construct and 3 concentrations. You have one buffer that's for the whole plate, and then you're just varying concentrations of different constructs. So you might set up a plate like this. If you're changing buffer and you might be doing a drug screen or a pH screen or a salt screen, it's the very same protein. It's just that the buffer is different. Um, you want to border your sample on both sides with a buffer because that buffer subtraction is really essential. So you got condition 1, buffer or sample in condition 1, and then condition 1 again. And then there's like a washing and all that stuff to make sure that there's cross-contamination, etc. Let's see. Oh yeah, I think that's similar. Okay. So controls in conducting an HT SAXS study. Again, you could waste a lot of samples if your sample is aggregated from the beginning, right? So do a small control first, send us a small plate. Yeah, if you do a full screen, it's just a lot of sample. So just don't, proceed cautiously. Check for concentration dependence. If you're doing one protein in a bunch of different conditions, run both. Run both HT and SEC SAXS. You can compare those results. Maybe you get exactly the same thing, but you might find things in SEC SAXS that preclude you from wanting to do HT SAXS or something like that. You can include in a 96-well plate a control like lysozyme or something in every plate. If you're doing a drug screening and you're using DMSO, we see that DMSO itself induces conformational changes. So make sure that you do a DMSO check. And then, we're going to check for solubility. We don't want clogged needles. We don't want crud up that hangs around on the windows. And, solid particles. If your sample's got dust in it or aggregates in it, it's going to drift through the beam. And we're improving this. We're trying to get smaller volumes, trying to replace needles quicker, temperature control, multiple plates on a deck. And again, we're upgrading the entire end station. To do this is the only beamline that you'll find this type of system on. And so we've gone through iterations of needles. This is the one that we're currently using. We've got Brandon has worked really hard on creating swappable needles. So like this one just inserts into the can and ready to go. We do have temperature control. This is brand new. This is all of these are 3D printed needles. So the needle can land into a temperature control device and you can ramp the temperature during exposure and collect data. It's lamentable that last time we collected, we didn't use this system, but I envision that we could do similar things to, differential scanning calorimetry, but with SAXS with this system. So like even for lipid nanoparticles, like how they open up as you change from 4 degrees to, room to body temperature or mutation stability. This is something I'd love to explore with people who have those kinds of problems. We're bringing down the volume right now. We're still 30 microliters, but we're moving towards real small volumes. This is 6 microliters in a plate. We'll see if this triggers or not. Seems like I'm slow here on this. Well, anyway, there's a small volume needle coming in and pulling up on 6 microliters. All right, trying to advance. Okay, so now you have a lot of data. How do you automatically analyze it? And creating an automated pipeline for every condition is very hard. Okay, so people aren't just studying protein, they're studying lipid particles, they're studying all kinds of things. They're studying how proteins feel each other in solution at long distances. Here's just some examples. This one has a structure factor to it. They're charged particles at a distance. How close do they get as you increase concentration? Here's another very small protein, but it's got a ton of aggregation at small q, so you want to chop this out. But how do you automatically analyze this in the same way you do that? Blue curve is a happy protein, and then you got some noise. So these are kind of how do you build a tool for that? And we're working on it. It's not perfect right now. A lot of it's about doing it by hand. Buffer subtraction is difficult, especially for things like lipid nanoparticles where you're trying to buffer exchange. And so this is sample and buffer. There's a small peak here that you can't see when you do the sample and buffer, but when you subtract the two, you get a nice peak, and that tells you about this looks kind of like a hexagonally packed lipid with some amorphous stuff. But you know, the buffer subtraction wasn't good, so you might have to adjust how much the buffer was subtracted by in order to get the right profile. And deciding what's right and what's wrong will take time. And this is, again, I just mentioned lipid nanoparticles. I didn't see anybody in the spreadsheet mention their particular interest in this, but I just wanted to show some SAXS profiles from lipid nanoparticles and some of the work we're doing there. So these are cryo-EMs from lipid nanoparticles. Each of these structures, phases, so you got lamella that are spaced just like a lipid membrane. Those have a particular peak distance. And then inside, deep in the core, there's some packing that's going on that's more hexagonal, and that gives a different peak. And because of the strength of those, you get extra peaks that are either a cube root of the first or square of the first, and that tells you whether it's lamellar or hexagonal. And so we were able to decompose these structures in a high throughput way because for lipid nanoparticles, the formulation space is huge, right? So all kinds of time and percent components and how it was mixed and all that stuff. So this is a big space to work on. Radiation damage. So when you receive your package in high throughput, you will get 30 frames. And those are because we're collecting for a full 10 seconds, but we're framing the data. Each of these blocks means we've put the data we collected through this interval into a separate file. And then you might imagine that over time, radiation damage is occurring and is affecting your sample. And we want non-radiation damage sample. And this is made possible by this new this detector we got about 10 years ago, this Pilatus. And we have a tool to help you with this. And this is how radiation damage will show itself. Largely in the small q, you will get changes. And sometimes in different regions, you might be able to accept only 2 frames here, 2 frames here. But over here, you might be able to take 10 frames. Everything's the same for a bunch. And then out in the high Q, yeah, you can take all of it, right? So it affects different regions in different ways. And we have a tool that separates these into regions and allows you to move how many frames per slice. And that's this, it's at our webpage and allows us to adjust these things. And you want highest signal to noise. So this is a protein that's not particularly radiation sensitive. And, the noise is just going back and forth. And the black curve with the, obviously has much smaller error bars than just one of these curves. And same with the high Q. Again, in terms of automated tools, we're developing things that sense this. So in this particular case, in the radius gyration region, it's stable for a while, and then the radius of gyration ticks up and then stays at a certain point. This is implemented here. So there's a light gray curve, a black curve, and a red curve. The light gray curve is the first sample. The black curve is all of these 6 samples or 7 samples. And then after that, if we include everything else, you get this red curve. So you just want as high signal noise as possible. Let's see if this will run. So you'll notice up here, something going back and forth. That's because we're trying to limit radiation damage by oscillating the sample inside the needle. But sometimes that creates high density and low density phases of the liquid, Sometimes you'll get a bubble. And so, sometimes it does causes more problems than not. So if you're having trouble, and you open up your scattering profiles and you get some kind of big oscillations, it could be because of our attempts here, and we're working on improving that. I'll skip this. Again, this is about structure factor data and high concentrations. Again, we want you to move through this as quickly as possible. We want you to extract the mass, radius, duration, maximum dimension, pair distribution function, and then get onto your modeling of the data as quickly as possible. And yeah, I think Mikkel will go through a lot of that stuff. One of the tools that we've developed, and I'm going to check my time here. Can't do that effectively. Is to do comparison measurements. So behind each of these boxes is a scattering curve. Okay. And in this particular box, we're comparing the scattering curve of the same thing to the same thing. It's a hexamer that has all of its multimeric units in the same conformation. But we have now, in using Fox, moved two of those subunits into a different conformation that's physiologically relevant. That SAXS curve has changed. And so we can represent that as a color, right? So these guys are the same, these are slightly different. And if we move this from a holo to an apo state, all subunits, the SAXS curves are very different. And we've represented that color as, devoid of similarity or white. Okay, so this is a sort of a matchup tool that's very easy to use. And we've shown that you can do this with mixtures. So this is the famous ATCase. So it's 100% in T state, but then you have mixtures, maybe 25% tense and 75% relaxed in a particular condition. Those will generate different SAXS curves, and you can see that in a very nice way using a metric of comparison between SAXS curves. Flexibility, something that's goes from a very flexible state to something that's very compact and rigid because of some modulation, and even conformational changes of the ribosome and the big and small subunit are swiveling and rotating relative to one another. Those are introducing different SAXS curves that can be compared against one another, and you can step through it. So this is a pictorial. It's probably worth demonstrating this. Maybe I'll go to the tool. Let me minimize this. Sorry, that's that. So I'm going to go to sibbles.lbl.gov. Here are our web apps. If I go to SAXS Similarity, this is a drag and drop tool. I've got my tutorial here. And so the particular one I'm going to demonstrate is each step of this morph. Okay. So I have examples there. So you can pull this down. I click that. I'm going to go to my folder where I have those scattering profiles that are each step of this morph. And what I've done for this morph is I have randomized the steps. So I've got randomized frame 0. And the reason I've done that is because I'm going to load it into this tool in a random fashion. All those steps are going to be random. So this is actually, this is randomized step 0, but it's frame 2 from that morph. Hopefully that makes sense to you guys. And I'm going to drag and drop this into, and Kevin, God bless his heart, was a fanatic for fancy and flashy things. So if I click on this, this is a randomized frame 4. It's actually frame 12 of the morph. If I click on this, I can see the SAXS profiles that compares randomized 4 frame 12 to the second one, which is randomized frame 3, but it's actually the 19th step in the morph. And I got those SAXS curves, and it gives you a radius of duration difference, the delta RG between them. And I can specify what Q range I want to compare into. So I might want to really focus in on, just this region. This is too noisy out there, and it'll change it. And then I can use other tools to help me cluster which ones are similar and which ones are different. So flashy graphic nicely organizes this, and you'll notice that the first frame 0, is at the bottom. That's the first step in the morph, and the last step in the morph is up here or close to. I can reverse that order so I get it in the right order. I can compare the most different curves against one another. So that's a super simple way of analyzing it. We can't really get much simpler, I think. We also have some other fancy things like this force plot. Again, not a lot of people have used it, but basically each SAXS curve is represented as a node. The size of the node is the same radius as, or is proportional to the radius of gyration. So in that morph, there's a compact state going to a large state. So this is the compact state, and then it expands to the bigger state. And the force between each of these nodes is how different they are. So you can see as this automatically goes through them. You can use different methods. So we invented this volatility of ratio. So it basically takes the ratio of two curves rather than the difference of two curves, which is what chi-square does. And different metrics of comparing the SAXS curves to one another will give you different results. So chi-square will kind of tweak it a little bit more in a nice linear way here. But if I go back to the curve and I use chi-squared, you see that it's a little less distinct, like it's a little blurrier as you go out. Anyway, that's the frame slice tool. Or sorry, the SAXS similarity tool. Let me go back to my presentation. Any questions about that? Let's see. So that's that tool. Yeah. So you might want to do this with proteins that have many metabolites. So this is the DNA repair protein called MUT-S. It binds ATP and converts it to ADP to do some mechanism. So we're interested in ADTP and ADP. We're interested in non-hydrolyzable AMP, PMP. This is the binding site up here. It binds DNA. So we might want to try different types of DNA. This recognizes mismatches. And so combinatorially, there's a bunch of different things that you might want to study. But there's all kinds of other issues. This protein is modular, so a different subunit can swap out and in. It might have different reaction to these things and these DNA. And then there's cancer mutations, right? Each of those mutants might be interesting to study in this way. So this frame slice tool is really helpful for stepping through the ADP, ATP, ATP gamma state, and with and without substrates, and trying to understand things that are similar. So yeah, so this is the ATP, you'll see ADP and ATP are very similar to one another. That's sort of this block. That means that it's hydrolyzing ATP very quickly to make it ADP. But then the non-hydrolyzable ones are very different. So it's stuck in a conformation where it's bound ATP, can't hydrolyze it. So MUT-S is stuck. And then you'll notice that these guys are over here. And that there's a diagonal of similarity here, which again has similarity to the apo state. So anyway, there's a lot of things that you can do and analyze that. We have created ways, and please come and do this at our facility. You can do 10 mils of culture, do a whole mutational analysis, and create mutants. And somebody's also done, I can actually name the user, Chris Brosey, who's now at a company, was doing an alanine scan on this particular protein. So it dimerizes, and she was trying to understand all of the amino acid chains along the way that communicate an event over here to the dimer interface. So she was scanning the whole protein with alanines and trying to identify the mutations that came across and eventually identify the chain, the pathway. And so if you can find the pathway, then you have all kinds of binding sites for small molecules to inhibit the dimerization. So I've demoed that guy. I think I'm going to stop there and see if there's any questions. Anything about HT-SAXS?
I've got a quick question.
Yeah.
So you had mentioned that using SAXS can help you determine if the protein will be a good candidate for HT-SAXS.
Yeah.
What would be signs from SAXS data that would suggest it's not going to be a good candidate?
This is Mikhail's favorite topic because I've taken charge of the HT so far, and he's taking charge of Mikhail of the SEC SAXS, and he always looks at his SEC SAXS and is like points out, oh, you've got some impurity here. There's like a little peak there, and how do you interpret the presence of increasing aggregates or dimer versus conformational change in the monomer? Like all of it's averaged together, right? So in HT-SAXS. So knowing that there's two species there and interpreting the data that looking at the data and asking, are the changes I'm seeing because there's a conformational change in the monomer, or because the percentage of dimer is increasing? You know, those are the kinds of things that will come out with your SEC SAXS data. Does that make sense?
Yeah, definitely. Thank you.
Yeah.
I would also add, I mean, if you're collecting data, there are insoluble proteins. So, and you really have some percentage of soluble proteins of your target. In SEC SAXS, you will immediately see that you have aggregations problem or some kind of ratio between not well-behaved and well-behaved samples. So the signal from the aggregated or larger species will overcome the signal from the smaller species in HT, and the differences that you're looking with and without ligand will be less visible. So you need to work on your, let's say, buffer conditions or purification steps.
Other questions or comments? Things that you guys wish I had covered better? I'm sorry if I missed it. You can use raw for analyzing HT-SAXS data. It just turns out that raw is particularly powerful for SEC SAXS data. But Primus and Scatter by Rob all work very nicely, and I use a combination of all of the above because all of them do different things better than others. But if you guys have questions, again, we hold weekly office hours. So if you collected the week before, come to office hour if you have a question. And also simple scattering, you can deposit your high throughput data there. Please do that. That's what it was originally designed for. It's just not as, you have to do it post, like you've analyzed all your data, you've got your radiation damage under control, there's no radiation damage sample, then you create your 40 SAXS curves and you deposit that into simple scattering, and it will give you a code. And then you, with your paper, you can say, okay, all the data is here. Good. Thank you, guys.